Good afternoon, students. Today we are seeing a topic our biomolecules. Bio means life and molecules mean the particle. Here the life is made up of our particles. See the 3D diagram of our biomolecules. In planet Earth having that is a wide diversity of living organism, means as in the previous lecture we saw that is the unicellular and the prokaryotic and the eukaryotic, that is the unicellular and the multicellular. Uni means the having the single cellular bacteria and multicellular including the plants and animals. Biochemistry, it is the branch of biology, understood student, branch of biological chemistry that provides us idea of chemistry of living organism and molecular basis for the changes taking place in plants, animal and the microbial cells, in which whatever the changes are taking on the basis of molecule, that is known as the biochemistry. There are mainly the 6 molecules that helps in the biochemistry, that is the carbon, hydrogen, oxygen, phosphorus, nitrogen, sulfur and the phosphorus. Planet Earth having a wide diversity of the living organism, in which they are having the east and including east or the multicellular including plants and animals. In biology, the biomolecules are divided in biology. Biomolecules are mainly divided into the 2 parts, that is the inorganic parts and the organic parts. In inorganic parts, minerals, gas, water are present and are in organic parts, that is the organic parts that contain the carbohydrate, lipids, amino acid, proteins, enzyme, nucleotides, nucleic acids, and the vitamins. In biomolecules, the micromolecules are also present and the macromolecules are also present. What is the micro? That means the very small quantity. Small size, low molecular weight between the 18 to the 800 Daltons and found in the acid soluble fuels, minerals, gases, water, sugars, amino acids, and the nucleotides. They are required in minor quantities, and micro means a large size, that is the high molecular weight about the 10,000 Dalton found in acid soluble food. They are carbohydrate, proteins, lipids, and the nucleic acids. In this living organism, there are mainly 3 types of macromolecule, which are the polymers of the subunit, that is called as the monomers. Mono means single and mers means the unit, means they are having the polymers, means the poly means the many, that is the many units are made up of single unit, that is known as the macromolecules, in which the polysaccharides, polypeptides and the polynucleotides are present. Saccharides, saccharin, that is the sugar, means polysaccharides means the carbohydrates and polypeptides means the proteins and polynucleotide, that is the nucleic acids. Nucleic acid which contains the DNA as well as the RNA. These are the polymers of the monosaccharides, amino acids and the nucleotides respectively. Monosaccharides means again the saccharin, that is it comes in carbohydrates. Then amino acids, that is they are the building blocks of our proteins. And nucleotides means they contain the genetic material, that is the nucleic acid, DNA and RNA. First, we will see one after one. In this topic, we are studying mainly the carbohydrate, protein, lipids and enzymes. Okay. In this, what is the carbohydrate? Carbohydrates are made from just three elements, that is the carbon, hydrogen and the oxygen, with the general formula CH2ON. Here N, that is the number of carbon. If we put the carbon, that is the 3, N is 3, then it comes the C3. C3 means the triads. We will see afterwards. Hydrogen and the oxygen are present in the same ratio as in the water, 2H2 As all of you know, water that is having the H2O formula, means hydrogen 2 and the oxygen 1. Depending upon the number of sugar unit, they are classified. See a broad classification of the carbohydrates, in which the carbohydrates are divided mainly into the three parts: monosaccharides, monomers, disaccharides, two saccharides, and poly means the many sugar molecules. Monosaccharide, it comes in simple sugars. Disaccharides, it comes in two monosaccharides. It is formed by the two monosaccharides, and poly means the many, that is which is nothing but a polymer of the monosaccharides. In monosaccharide, trioses, as the name indicates, trioses, tetroses, pentose, hexose and heptose. Triose, 3 carbon compounds, tetrose, 4 carbon, pentose, 5 carbon, hexose, 6 carbon and heptose, 7 carbon. Here N is equal to 3, 4, 5, 6, 7. Accordingly, the number of hydrogen and oxygen will be changed. Means, see the example of the trioses is the glyceraldehyde. Example of the tetroses, it is the erythrose. Example of the pentose, it is the RNA, ribose in RNA and deoxyribose in DNA. Then hexose, that is the glucose, that are having the 6 carbon compound, and it is observed in blood sugar. And fructose, that is observed in fruit sugar. And galactose, that is nothing but the product of the lactose. And heptose, it is made up of the 7 carbon. Disaccharide, it is made up of 2 monomers or the 2 monosaccharide. Disaccharide, it is formed by the fusion of 2 monosaccharides. Example, sucrose, it is found mainly in the cane sugar, sugarcane juice. On hydrolysis, that is, it produces glucose and fructose. If we remove the water from sucrose, it converted into the glucose and fructose. In lactose, that is mainly present in milk sugar, on hydrolysis, it produces glucose and galactose. And maltose, another example of the disaccharide, malt sugar, on hydrolysis, it produces 2 units of the glucose. By seeing polysaccharides, there are mainly the 2 groups, that is the homopolysaccharide and the heteropolysaccharide. Homo means that is a polymer of one type of monosaccharide. Example, starch, that is the plant storage molecule. And cellulose, cell wall component. Example, glycogen, animal storage molecule. Starch is present. Starch is, that is the stored food material in plants. Cellulose, that is mainly the cell wall components in the plant cell and animal cell. And glycogen, that is the animal storage molecule. Cellulose is mainly present in the plant cell only. Okay. Then heteropolysaccharide, in which that is the polymer of different types of monosaccharide, which is made up of not the same monomer, that is a different higher uranic acid, heparin and blood group. Classification of carbohydrate. First one, monosaccharides. Mono means single, saccharides means the sugar. Monomers, these are the building blocks or monomers of the complex carbohydrate. These are the simplest sugars having a crystalline structure. They have general formula CH2ON. where N can be, as I said, can be triose or tetrose or pentose or hexose or heptose. Okay. Mono means single. They are the mainly building blocks of complex carbohydrates. Then monosaccharides are further classified as glucose, fructose, and the galactose. What is the glucose? Most important fuel in living cells. Student, as you know, if any student has fed up, we give you, that is the glucose solution, as glucose is directly absorbed into the blood. It is present about 90 milligram per 100 ml of blood. It is produced because of cellular respiration. Inside the cell, the respiration occurs and glucose is produced. C6H12O6, that is the structure of the glucose. And by photosynthesis also a glucose is formed. I hope all of you understood the glucose. Second one, galactose. It is a similar molecules like the glucose, consists of alpha and beta particle. Same like glucose, but having a difference in alpha and beta particles. Galactose react with glucose to form the disaccharide lactose, and it does not form by the cellular respiration. Means glucose monomer, galactose monomer, they unite and form a disaccharide lactose. All of you know the fruits having a fruits are in sweetened taste. This is the sweet taste produced by a fructose. It is the fruit sugar and chemically it is called as ketohexose. And fructose reacts with the glucose to form the sucrose, which is the disaccharide. Students, see here, fructose reacts with the glucose to form the sucrose, and sucrose, that is the disaccharide. Fructose, it is the similar to the glucose. Now, going ahead, disaccharides. What is the disaccharide? The saccharide is formed when two monosaccharides react with each other. Two monosaccharides unite and they form the disaccharide. Example, sucrose, lactose, maltose, etc. A bond is present, that is known as the glycosidic bond, forms and holds the two monosaccharide units together. The overall process of the formation of disaccharide, it requires the energy. Example of disaccharide, that is the sucrose, lactose, and the maltose. Then sucrose is called as the non-reducing sugar since it does not have the free aldehyde or the ketone group. It cannot be reduced, means they cannot having a free aldehyde, that is a CHO group, and the C double bond O, that is a ketone group. Lactose and maltose are the reducing sugar, and disaccharides are soluble in water. As we know, the sucrose, that is the, it can, if sucrose is dissolved in water, it can easily mix with the water, that is the soluble in water. And disaccharides on hydrolysis, if the water is removed from the disaccharide, it forms the monosaccharides. Then third class of the classification of carbohydrate, that is the polysaccharides. Poly means many, saccharides means the having the many monomers. When monosaccharides undergoes a series of condensation reaction, adding one unit after the another till a very large molecule is formed. In starch, there are many glucose molecules are added. There is a long chain of the monosaccharide. This is called as the polymerization. And polysaccharides on hydrolysis gives a monosaccharides. Then starch is the one example of the polysaccharide. Starch is a type of stored food in the plants. As all of you know, in the leaves of the plant there is a food material, that is the starch. Again, it exists in two forms, that is the amylose and amylopectin. Starch having the mainly two parts, two forms, that is the amylose and amylopectin, in which the unbranched polymer is known as the amylose of alpha glucose, and amylopectin, it is the branched polymer of the alpha glucose. Unbranched and the branching, it is the mainly difference in amylose and amylopectin. They are structurally different to the each other. Then glycogen, it is the type of amylopectin with very short distance between the side chains. It is present in the human and the animal's body, especially in organs like the liver or muscles. In liver and muscle, the glycogens, live muscles of the animal body, the glycogen is present. If we are starving, then this glycogen get converted into the glucose. And next important part of the starch, next important part of the polysaccharide, that is the cellulose, and this type of polymer made from the beta glucose molecules. Cellulose is generally present in cell wall of the plant cell. These are stronger, tougher than the other polymers. This toughness is generally due to the hydrogen bonding between the neighboring chain. Cellulose, where it is present in the cell wall. Cell wall is having only the plant cell, and due to the cell wall, due to the cellulose, cell wall getting a toughness, a rigid structure to the plant cell, and cellulose, it is made up of the, that is the polysaccharide. Now see, what is the biological importance of the carbohydrates? First one, it supplies the energy for metabolism. We required a lot of energy for the metabolism, and this energy was given by a carbohydrate. Glucose is the main substrate for the ATP synthesis. Lactose, which is present in milk, provides to the infant babies. Polysaccharides helps in the structural component of the cell membrane and cell wall at the reserved food material as a starch. See, as I hope all of you understood the structure of carbohydrate, in which we saw that is the mainly the monosaccharide, disaccharides, and the polysaccharides. Now, second important parts of the biomolecule, that is the lipids. These are long chain hydrocarbon containing hydrogen, oxygen, and carbon. Lipids having the greasy consistency. All of you seen the oil and fats, they are having a oily greasy consistency. The hydrogen to oxygen ratio is greater than 2:1, and they are not soluble in water. They are soluble in only the organic solvent, and lipids are also termed as the fatty acids. What is the fatty acids? See, fatty acids are the organic acids, okay, in which are composed of the hydrocarbon chain ending in the carboxyl group. They may be the saturated or unsaturated. Saturated, in which the carbon having single bond, palmitic and the stearic acid found in animal and the plant fats are the example of the saturated fatty acids, in which what is the fatty acid example, that is the palmitic and the stearic acid, and many more fatty acids are present, and this organic acid that is formed by the hydrocarbon chain ending in the carboxyl group. Unsaturated fatty acid, the difference in the saturated and unsaturated, that is having the stability. Saturated means they are more stable as compared to the unsaturated fatty acids. In these unsaturated fatty acids, one or double bond between the carbon atoms of the hydrocarbon chain are present. Oleic acid is found in all fats, and the linoleic acid, it is found in many seed oil, are the examples of the unsaturated fatty acids. See, oleic acid, it is found in all fats, but the linoleic acid, it is found in many seed oils, are the example of the unsaturated fatty acid. Lipids again classified as similar to the carbohydrates, simple lipid, compound lipid, and the derived lipids. Simple lipids, what are the simple lipids? These are the esters of the fatty acids with the various alcohols. Fats and waxes are some of the examples of the simple lipids. Fats are the nutritional sources with the high calorific value. Also, fats acts as the reserved food material. Simple lipids म्हणजे काय? Esters of the fatty acids with the various alcohol. Fats and waxes. Waxes, all of you know the candle, that is the wax, which is found in a solid form. Then in plants, it is stored in seeds. In seeds, the lipids are stored, and in animal fats, it is stored in the adipose tissue. In fat deposited in tissue acts as an insulator and minimize the loss of body heat. Fat deposited around the internal organs act as the cushions to absorb the mechanical shocks. Plants getting some mechanical shocks, and due to the fats, it absorbs the mechanical shocks and the organ remains healthy. That is that fats are present in our visceral organs like the heart, kidney, brain, etc. Waxes. What is the waxes? Waxes are the esters of the long chain fatty acids with the long chain alcohol. See the structure. Waxes are the esters of long chain fatty acid. Fatty acids are present, and not only the fatty acid, esters of the long chain fatty acid with the long chain alcohols are present. Waxes are generally solid at the room temperature. All of you know there is a wax layer coating on the lotus leaves. Waxes form a coating on hair and skin in animals and also form the outer coating of the stem, leaves, and the fruits. Second one, going out ahead, second one, that is the compound lipids. These are the esters of fatty acid containing phosphates, that is known as the phospholipid. Phosphate group is attached to the lipid, that is known as the phospholipids. Second one, if the sugar is attached to the lipid, that is known as the glycolipids. And in this compound lipid, the phospholipids and the glycolipids are present. These lipids consist of a molecule of glycerol and two molecules of fatty acids and the phosphate group. Students, in compound lipids, they are not like a simple lipid. They are having a group of phosphate and lipid attached to it, that is known as the compound lipid. In glycolipids, they contain the glycerol, fatty acids, simple sugars such as the galactose and the nitrogenous bases. They are called as the cerebrosides. What is the cerebrosides? In which of fatty acids, glycerol, and which contains the which are in the glycolipids, and simple sugars such as the galactose and nitrogen bases. They are called as the cerebrosides. Cerebrosides are related to the brain, and these are the compound lipids. Simple lipid plus any other group, if it is attached like the phosphate group, they are known as the compound lipids. Third one, derived lipid. Derived lipid, it is the nothing but it is the form. It is formed by the lipid. Means they are composed of fused hydrocarbon rings and a long hydrocarbon side chain. That is, they are the sterols. Students, sterols composed of fused hydrocarbon rings. Hydrocarbon ring, that is the CHO rings, and long hydrocarbon side chain. They have very complicated structure, that is known as the example cholesterol. It is mainly present in all cells of the animal's body, but particularly in the nervous tissue. Example, our hormones, that is the adrenocorticoids, sex hormones, in which the progesterone, testosterone, and the vitamin D are synthesized by the cholesterol. See the important function of the lipid, that is the derived lipids, in which the cholesterol, they are helpful to form the hormones. In young plant, that is the Dioscorea, a steroid compound called diosgenin is produced. It is used in the manufacture of the anti-fertility pills, that is the birth control pills. Lipids, in lipids we saw that is the simple lipid, derived lipid, and the compound lipid. Going ahead, the main third biomolecule, that is the proteins. Proteins are known as the body building compound. Proteins are very helpful for our structural development. Proteins are the large molecule containing amino acid units ranging from 100 to the 3000. In proteins, amino acids are linked together by the peptide bonds. The peptide bonds are present in proteins and they are linked to each other. A protein molecule consists of one or more than one polypeptide chain. These proteins can contain any of the 20 natural occurring amino acids type. Proteins like the keratin of the hair, there are essential 20 amino acids are present. Okay, in which some example are cysteine, then leucine, methionine. These are the example of the essential 20 amino acid. Proteins like keratin of hair, in hair keratin protein is present, which consists of polypeptide chain arranged like a spiral helix. In some proteins, 2 or more polypeptide chains are linked together by the intermolecular hydrogen bonds. Means proteins, they are formed by the amino acids, and amino acids are linked each other by a peptide bond, in which 2 or more peptide chains are linked together by intermolecular hydrogen bond. Such structure are called as, that is the pleated sheet, and this pleated sheet structure is found in the protein of the silk fibers. In large proteins such as the myoglobin, hemoglobin, myoglobin, these are the example of the protein. The large protein myoglobin, peptide chains are looped, twisted, and folds. In the structure of myoglobin, the peptide chains are looped. They form the loop as well as they get the twisted, as well as they are having a folded nature. In protein, they are extremely reactive and the highly specific in behavior. Proteins are the amphoteric in nature. They are acid as well as the basic. That is, they can be survived. That is, they are having the both the nature, amphoteric in nature. On the basis of structure, proteins are divided, classified into 3 categories, like the simple proteins, conjugated proteins, and the derived proteins. Similar to the lipids, proteins are also divided mainly into the simple proteins, simple lipids, conjugated proteins, conjugated lipids, and derived proteins and the derived lipids. Now, simple proteins. Simple proteins, they are soluble in water, and on hydrolysis, proteins are formed by the amino acids. So on hydrolysis, they give the amino acids. Example, myoglobin, as I told. Then next one, albumins, and they are present in egg white. Albumin is present in egg white, and these albumins are soluble in water, but they get coagulated on heating. What mean by coagulation? They form the precipitate and on heating. Albumins are widely distributed. Example, egg albumin, serum albumin, and legumelin of pulses are the albumins. Legume plants like the methi, they are having a albumin, legumelin. Leguminin of the pulses are the albumins. These albumins are mainly present in the eggs. Then conjugated proteins. Conjugated proteins again consist of the simple proteins combined with the some non-protein substance. Means there is a simple protein is present as well as the non-protein substance is present. This non-protein group is called, that is the prosthetic group. Students, some a non-protein group that is called as the prosthetic group. Example, hemoglobin. Heme plus globin. Okay, heme that is not having a protein group, that is known as the non-protein group, prosthetic group. These proteins are classified as glycoproteins and the mucoproteins. Mucoproteins are the carbohydrate protein complexes. Example, mucin of the saliva. And lipoproteins are the lipid protein complexes. Example, plasma membrane. Third one, derived protein. This type of protein is not practically found in nature. Means it is formed in the lab. These proteins are derived from the native proteins molecules on hydrolysis. The example, metaproteins and the peptones, etc. Going ahead, as we see the basic three components, that is the proteins, carbohydrates and the lipids and their classification. The next important components of the biomolecule, that is the nucleic acid. Nucleic acids are nothing but the genetic material, that is the DNA and RNA, and this genetic material are present in all the cells. Nucleic acid, what is the nucleic acid? Nucleic acid are the biopolymers or large biomolecule essential to the life. Nucleic acids are of two types. First one, DNA, deoxyribose nucleic acid, and RNA, ribose nucleic acid. DNA, deoxyribose nucleic acid, and RNA, that is the ribose nucleic acid. These nucleic acid contain two pyrimidine. See here, there are pyrimidine bases are present as well as the purine bases are present. Pyrimidine, that is the cytosine and thymine, and the purine bases, that is the adenine and the guanine. Understood? See here, there is some trick like the AGP. What is the AGP? Adenine, guanine, purine. It forms the, where the adenine and guanine is present, that is the purine base. And if the CTP, that is the cytosine and the pyrimidine, cytosine and the thymine is present, that means it is the having a pyrimidine base. Then DNA, it is composed by sugar molecule, phosphoric acid and the nitrogen bases. DNA mainly composed by three molecules: sugar molecules, phosphoric acid and the nitrogen bases, in which principally bases are two types, as I told, pyrimidine bases and the purine bases. Pyrimidine bases are the single ring nitrogenous bases. They are having a single ring nitrogenous bases. Example, cytosine, thymine, uracil are the pyrimidine bases. And in purine, in pyrimidine single ring nitrogen bases is present, but in purine double ring nitrogen bases are present. Example, adenine and guanines are the purines. Then now see the structure of DNA, as DNA, that is the genetic material present in our cells, and DNA, which is a very vital nucleic acid, it is present in our nature at very extent manner. Structure of DNA. DNA is a very long chain made up of alternate sugar and the phosphate groups. This sugar is always deoxyribose sugar. Then this unit, which consists of sugar, phosphate and base, is called as the nucleotide. See student, what is a nucleotide? Sugar, phosphate and nitrogen base. That is the nucleotide. And nitrogen bases and a sugar of nucleotide, it forms the nucleoside. In a nucleoside, nitrogen bases is attached to the first carbon first carbon atom of the sugar. When a phosphate group is attached with the carbon 5 atom, it is a C5 atom of the sugar molecule, a nucleotide molecule is formed. When the nucleotide molecule is formed, phosphate group attached with the carbon 5 atom of the sugar molecule, and a single strand of the DNA consists of the several thousand nucleotides, one above the other. A single polynucleotide strand of DNA is not straight, but it is the helical in shape. See the structure of DNA that is the, see, it is not a single strand structure, it is the double helical structure, and they are nitrogen bases are attached in this helical structure. 3.4 nanometer, that is the one distance between the two helix, one helix. See, the DNA molecule consists of such a two helical polynucleotide chain, and the two complementary polynucleotide chains of DNA are held together by a hydrogen bond, and they are weak in nature. Adenine always pair with the thymine, a double bond thymine, and guanine, it is with the cytosine. That is, pyrimidine bases are filled with the purine bases. Okay, all of you understand the DNA. See, double helical structure and structure of DNA. Going ahead, ribonucleic acid, that is the RNA. Like a DNA, ribonucleic acid, it is also consists of the polynucleotide chain, with the difference that it consists of a single strand. Means in DNA, they are having a double stranded structure and helical structure, but in RNA, they are having only a single stranded structure. The nucleotides of the RNA have the ribose sugar instead of the deoxyribose sugar. Students, in DNA, deoxyribose sugar is present, but in RNA, ribose sugar is present. In case of the RNA, uracil substitutes of the thymine of DNA. RNA means in RNA, uracil substitutes thymine of the DNA. In DNA, thymine is present. At that place, uracil is substituted. And most of RNA polynucleotide chain starts either with the adenine or guanine. In RNA, they are formed by the adenine or guanine. In RNA, there are mainly the three subtypes that is known as the mRNA, rRNA, and tRNA. mRNA, what is the mRNA? It carries the genetic information for arranging the amino acid. mRNA, that is the messenger RNA, which carries the message for the protein synthesis. It accounts 3% of the cellular RNA, and size of mRNA is related to the size of message it contains. And synthesis of the mRNA begins at 5' end of the DNA and ends with the 3' end of the DNA. Then rRNA, that is the ribosomal RNA. These ribosomal RNA are attached with the ribosomes in the cell. In rRNA, they form 50% to the 60% part of the ribosome, and it accounts mRNA that counts the 3%, rRNA it accounts the 80% to the 90% of the cellular RNA, and it is synthesized in the nucleus. Then tRNA, that is a transfer RNA, that this transfer RNA, tRNA molecule are such a smaller, consisting of the only 70 to the 80 nucleotides, and they are having a loop structure. And in the anticodon loop of tRNA, 3 unpaired nucleotides are present, that is are called as the anticodon, which pair with the codon present on mRNA. This tRNA having a hairpin like a structure. Okay, in nucleic acid we are seeing the DNA and RNA. In DNA, the structure of DNA is observed, and in RNA we are seeing a structure of mRNA, rRNA, and tRNA, and they are all are helpful in they carry the function of protein synthesis. In going ahead, the part of the our chapter, that is the enzyme. Enzymes are nothing but the biocatalyst. What is the biocatalyst? Students, here biocatalyst means they cannot participate in the reaction, but they accelerate the rate of reaction. As in exam, we cannot help to you, but by learning you or by teaching you, we accelerate your rate of study. Like this, enzymes are the biocatalyst. Like the teacher also the biocatalyst, which speed up a chemical reaction. If these enzymes were not present in the cell, either the reactions would not occur, or if they occur, they would occur at very slow rate. Means enzymes having very vital role. If they are present, then the reactions are going ahead or reactions are started. Now, otherwise it forms at very slow rate. The substance upon which the enzymes acts, that is termed as the substrate. Means the substance on which the enzyme acts, that is known as the substrate. The enzymes which are present in the cell are known as the endoenzyme. Endo means inside. The enzymes which are present internally in the cell or internally in the cell, that is known as the endoenzyme. And enzymes, example, enzyme produced in the chloroplast and the mitochondria. Chloroplast, mitochondria are present inside the cell. Enzymes are produced inside the mitochondria and chloroplast, means that is known as the endoenzyme. If the enzymes are present outside the cell, that are called as the exoenzyme. Example, enzyme released by the many fungi. Then now see the nature of enzyme. Depending upon a chemical composition of enzyme, it is of two types. Purely, first one, how the nature of the enzymes are there. First one, purely proteinaceous enzymes. Example, protease that spills the protein. And second one, conjugated enzymes, means they are which with the non-protein prosthetic group is attached. First one, purely proteinaceous enzyme, means they are having a pure protein structure. And second one, that is the conjugated enzyme, simple protein, simple enzyme plus non-prosthetic group, that is the conjugated protein. In purely proteinaceous enzyme, example, that is the protease, and in conjugated enzyme, example, that is the NAD, FAM, etc. Here, the organic compound that are tightly attached to the protein part, that is called as the coenzyme. Means the enzymes is present and plus the organic compound, they are tightly attached to the protein part. Example, that is the NAD. What is the NAD? Nicotine amide adenine dinucleotide. And FAM, that is a flavin mononucleotide. Whereas the inorganic ions which are loosely attached to the protein part, they are known as the cofactors. Students, here in cofactors, inorganic ions are attached, but in coenzyme, the organic compounds is attached. Cofactors, inorganic ions, and in coenzyme, the organic compounds are attached, tightly attached. Example, magnesium, copper, zinc, iron, manganese, etc. And iron is the cofactor of enzyme. Metal cofactors are referred to as the enzyme activators. Means metal cofactor, they are attached to the enzyme activators. Now see the properties of the enzyme. First one, the proteinaceous nature. All enzymes are basically made up of protein. Means all enzymes are proteins, but all proteins are not enzymes. Means all enzymes, they are having a basically made up of the protein structure. Then three-dimensional shape. All enzymes have a specifically three-dimensional shape, that is the 3D structure. Then Next one, catalytic property. Enzymes are the biocatalyst, means they speed up the reaction or they accelerate the rate of reaction, but they remains unchanged. They cannot take part into the reaction. Next one, specificity of the action. Each enzyme acts upon a specific substrate or a specific group of substrate. Means each enzyme having a specific substance. They act on a specific substrate. Second one, that is the reversibility of the action. Enzymes are very sensitive to the other factors like the temperature and the pH. Any increase or the decrease in pH cause decline in the enzyme activity. Example, enzyme pepsin, which is secreted in stomach. Pepsin, which is the digestive enzyme, shows highest pH of 2, that is the 2 pH means the acidic pH, and trypsin, which is present in the duodenum, having the pH of 9.5, that is the alkaline. Temperature, enzymes are not active at the high temperature. Very high temperature means they are destroyed at 60 to 70 degrees Celsius. Now see how the enzymes are named, how the nomenclature of enzymes are done. Enzymes are named by adding the suffix, that is a ase, to the name of the substrate on which they act. Example, protease, here ase, that is a suffix is used for the enzyme protease. Then sucrase, ASE, here is the suffix that is used to nomenclate enzymes. Nuclease, ASC, the enzyme that can be named according to their type of function they perform. Example, dehydrogenase, remove the hydrogen. Carboxylase, add the carbon monoxide, and decarboxylase removes the carbon dioxide, and oxidase means the helping in the oxidation, that is the addition of the oxygen. See, dehydrogenase, I am repeating again, dehydrogenase, removal of hydrogen. Carboxylase, adding of carbon monoxide. Then decarboxylase, D means removal of carbon dioxide, and oxidase means the addition of oxygen. According to the International Code of the Enzyme Nomenclature, the name of each enzyme ends with an ASE and consists of the double name. Means they are having the two words and ends with the ASE. The first name indicates the nature of substrate and the second name indicates the reaction catalyzed. First name, nature of substrate. Second name, reaction catalyzed. Example, pyruvic decarboxylase. Pyruvic, that is a name of substrate, nature of substrate, and decarboxylase, that is a which reaction is catalyzed. Catalyzes the removal of CO2 from the substrate pyruvic acid. Now see the classification of enzyme. First one, oxidoreductase. These are the enzyme catalyzing oxidation and the reduction reaction by the transfer of hydrogen and or oxygen. Example, alcohol dehydrogenase. Transferase. These enzymes catalyze the transfer of certain groups between the two molecules. Example, glucokinase. Means they transfer the certain groups. Then hydrolase. These are the enzyme catalyzes the hydrolytic reaction. This class includes amylase, protease, lipase, etc. Example, sucrase. Next one, lyase. These are involved in the elimination reaction resulting in the removal of a group atom from the substrate molecule to leave the double bond. Example, fumarate. Isomerase means some enzyme catalyze the structural rearrangement within the molecule. Their nomenclature is based on the type of isomerism. Thus, these enzymes are identified as, that is a racemase, epimerase, isomerase, etc. That is a xylose isomerase. Ligases or the synthetase. These are the enzymes which catalyzes the covalent linkage of the molecule utilizing the energy obtained from the hydrolysis of energy-rich compound like the 70S like ATP, GTP, example glutathione synthetase, pyruvate, decarboxylase, ligases. Oxidoreductase here, which are the types of classification of enzyme. First one oxidoreductase, second one transferase, third one hydrolase, fourth one lyase, fifth one isomerase. Ligases means, students, you have to learn very carefully of this classification of enzymes, and these are very useful. And last one, the lyases. Then see the mechanism of the enzyme action. How the enzymes are act. What is the mechanism behind the enzyme action? In this enzyme action reaction, they are having a substrate as well as the enzyme. The basic mechanism by which the enzyme catalyzes the chemical reaction starts with the binding of the substrate. The binding of substrate to the enzyme causes the reaction that lead to the formation of the product. See, here the substrate is present and here the enzymes are present. These substrates are active. This substrate is reacts with the active sites of the enzyme, and it changes of the shape and to being a substrate. That is known as the ES complex, enzyme substrate complex. See here, substrate is present and enzyme is present, and it changes accordingly. Next, these enzymes mainly depend upon two models. That is the first one, lock and key model, and second one, induced fit model. Here the enzyme, it acts as the lock, and key, that is the substrate or the analogy, in which, see here, enzymes, active sites of enzymes are present on which the substrate, here the sucrose is the enzyme is present, sucrase is the enzyme is present, and this substrate act on this enzyme and they form, that is the enzyme substrate complex. A lock is the enzyme and the key is the substrate, and they form the reaction. Again, enzyme remain as it is the biocatalyst and glucose. So on the sucrose, that is the substrate, that is converted to the glucose plus fructose with the help of enzyme sucrase. Means they undergo the hydrolysis. Next, induced fit model. This model states that first one, in lock and key model, enzyme is the lock and key, that is the substrate, key, that is the analogy, in which the sucrose is converted to the glucose and fructose with the help of sucrase enzyme. Means the enzyme acts on a enzyme remain as it is, and it substrate is converted to the product, and it undergoes the hydrolysis. Example. Second one, that is the induced fit model. Which is the induced fit model? This model states that the approach of a substrate induces a conformational change in enzyme. In lock and key model, enzyme remain as it is, but in induced fit model, it forms a conformational change of enzyme. In this induced fit model shows that enzymes are rather flexible structure in which the active site continuously reshaped by its interaction with the substrate until the substrate is bound to it. Means enzyme having a various active site and they are changing as getting a substrate completely bound to it. They are changing their shape. Student understood? I hope you understood the lock and key model and the induced fit model. In lock and key model, enzyme is lock and key, that is a substrate, but in induced fit model, enzyme, that active sites of the enzyme, that is continuously changed to form to get substrate completely bound to it. There are conformational changes. Now going ahead, factors affecting the enzyme activity. First one, the concentration of substrate. Second one, the enzyme concentration. See, substrate concentration increases then the velocity of enzyme activity will be increases within a limited range of substrate level. Means that is the optimum level is substrate concentration is increases, activity of enzyme is also increases, but in limited period. Second one, enzyme concentration. The rate of enzyme reaction is directly proportional to the concentration of the substrate. Enzyme reaction is directly proportional to the rate of substrate. Enzymatic reaction. Substrate is increases, then again the enzyme concentration will be also increases. Then next one, temperature. The enzymatic reaction occur at the optimum temperature, that is around 37 degrees Celsius. At high temperature, it get denatured. In low temperature, it is inactive. Means they are best at around the 37 degree Celsius. Enzyme rapidly denatures at the temperature about the 40 degrees Celsius, as I told to you. Then effect of pH, there is also if pH at which the enzyme will catalyze the reaction at the maximum rate. Every enzyme has the different optimum pH value. Now see the other substances. The enzyme action is also increased or decreased in the presence of some other substances such as the coenzymes, activators, and the inhibitors. Coenzyme that helps for the enzyme activation, activator that activate, that inhibitor that stop the enzymatic action. Activators are the inorganic substances that increases the enzyme activity. Then secondary metabolites. Secondary metabolism are the small organic molecules produced by organisms that are not essential for their growth, development, and the reproductions. What is the secondary metabolites? Means the small organic compound produced by the organism, but these organisms, these secondary metabolites are the extra molecules produced by the cell for their growth, and they are not essential. Then several types of bacteria, fungi, plants produce the secondary metabolites. These secondary metabolites can be classified as terpenes, phenolics, and the nitrogen containing compound. Terpenes, they are made from the mevalonic acid that is composed of mainly the carbon and hydrogen. Phenolics, that is made from the simple sugars containing benzene ring, hydrogen, and the oxygen. And nitrogen containing compound, extremely different class, may also contain a sulfur. These terpenes and phenolics and nitrogen containing compounds are not required for the cell, but these secondary metabolites are useful for the these secondary metabolites are useful for the cell. Then see students, in this topic we are studied mainly the carbohydrates, then proteins, lipids, nucleic acids, in which the nucleic acids, DNA, RNA, structure of DNA, structure of RNA, classification of RNA, that is the mRNA, rRNA, and the tRNA. Then the enzymes, in which the nomenclature of enzyme, classification of enzyme, mechanism of action of enzyme, and then the secondary. In this mechanism of action of enzyme, so two mainly the mechanism, that is a lock and key model and the induced fit model. And next one, that is a secondary metabolite. Questions can be asked in the exam, so study carefully, and if you have any doubt, ask to me on my personal WhatsApp number. Then okay, we are stopped hearing. Thank you very much.
Thank you, ma'am.